Hello and welcome to Year 12 Biology. Today we're going to be going through non-Mendelian inheritance patterns. So we start with a bit of a revision. Remember that Mendelian inheritance is the kind of straightforward inheritance we've been talking about, where one allele is dominant and the other one is recessive. And if you inherit one of those dominant alleles, the recessive one is going to be hidden or carried. And this was originally observed and documented by Gregor Mendel, and so hence the name Mendelian inheritance. Today, what we're really focusing on is the rule breakers, the ones that don't follow this dominant and recessive. They do some special things all of their own. So the first example or type of non-Mendelian inheritance we're going to go through today is incomplete dominance. Then we're going to talk about codominance and the difference between the two. And then we're going to focus on sex-linked inheritance. Later in the week, we're going to do example 4, 5, and 6, which is multiple alleles or polygenic inheritance, lethal alleles, and pleiotropy. So let's get into it. Incomplete dominance is when one allele doesn't fully dominate over the other. The two are going to blend to create a brand new phenotype that is an intermediate of the two parents. So if we have a red flower and we're crossing it with a white flower, all of the offspring are going to be heterozygous and they're going to be pink. Now, I hate when they do this and they represent them as one big and one capital and one lowercase letter. The lowercase letters are supposed to represent the recessive alleles. So this is actually wrong. I would definitely not represent it with big R, little r. I would represent it as big R, big W. So this one would be big R, big W, big R, big W, big R, big W, big R, big W. Okay, both alleles get represented with capital letters. Otherwise, having a look at that diagram, how would you be able to tell without the pictures which ones are dominant and which ones are recessive, or whether they are incompletely dominant? It's almost impossible to tell from that annotation. So that's why we give them both capital letters. So let's do a worked example for incomplete dominance. This is the kind of question that you would get on a test. The allele for color in snapdragons is incompletely dominant. Snapdragons are not as exciting as they sound. They're not dragons. They're a type of flower. If a homozygous red flower is crossed with a homozygous white flower, use a Punnett square to predict the phenotype and genotype of the offspring. Okay, so first thing we've got to go back and do a little bit of vocab. We've got homozygous red flower. Homozygous, homo meaning same. So I have two red alleles. And it's crossing with a homozygous white flower. So I have two white alleles. Now, in a question like this, which one would I call the father and the mother? It doesn't actually matter where you put them, as long as in the end they all get included. So I'm going to put my white one up the top and my red one down the side. And I would end up with RW, 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 RW. So, what the question is asking me to do, I've got to go back. Oh yes, I've done the Punnett square, but have I answered the question? Use a Punnett square to predict the phenotype and genotype of the offspring. So there is 100% chance that the genotype is going to be big R, big W. They're going to get one of each of these incompletely dominant alleles. And it's 100% that the phenotype is going to be pink, halfway between red and white. We're going to get a new colour come out. Your turn. I want you to have a go at doing this one. So pause the screen here, read through the question, and we will go through the answer together. Okay, hopefully you've had a chance to do that now. Let's do it together. If two of the offspring from the previous problem were mated, okay, so I've got R, big W. Use a Punnett square to determine the genotype ratio and the phenotype ratio. Okay, so let's put big R, big W, big R, big W. So I have two heterozygous offspring that are now becoming the parents. So this offspring would get a big R from father and a big R from mother. So he's going to be red, or this flower is going to be red. This one's going to get a big W from father and a big R from mother. This flower is going to be pink. This one's going to be pink as well, big R, big W. And this one is going to get a big W from father and a big W from mother. So it's going to be a white flower. So I want the genotype ratio. Genotype ratio. So GR stands for genotype ratio. I've got 3 different genotypes here. I have big R, I have big R, big W, and I have big W, big W. So my ratio is I've only got 1 big R. I've got 2 of the heterozygous big R, big W, and I only have 1 homozygous white. So my ratio is 1:2:1. My phenotype ratio is going to be the same because this one is red, this one is pink, this one is pink, this one is white. So it's going to be red is to pink is to white, 1:2:1. Hopefully you got that answer. It's really important when you annotate these for the phenotype and genotype ratio that you define which alleles and which phenotypes you're talking about and the order. Because if you just give me numbers, I don't know which ones you're actually talking about. Really important that you determine those before you put your answer down. Okay, second form of naughty rule-breaking inheritance is codominance. And codominance and incomplete dominance can look really, really similar. So it's important that you pay attention in the question to how it's expressed and what keywords are in the question. In codominance, both alleles are completely dominant. They're both going to be expressed at the same time in the offspring, but in equal and different ways. There's no actual blending of the phenotype at all. You just get the same amount being expressed. So each and every cell is then going to have to effectively make a decision about which of these genes it's going to target and express. So instead of having a white cow cross with a red cow and you get a halfway between red and white, almost pink looking cow, you get what's called roan, where some of the hair cells are red and some of the hair follicles are white. So the fur ends up being red and white spotty. We represent both of the alleles again using capital letters. So some examples from the real world. We have roan cows. So if you've never seen a roan cow, you get a white father or a white mother, cross it with another red cow, and you end up with this phenotype that is a mix of the two. So you get some of the cells being red and some of them right next to each other. The next hair will be red and the other one will be next to it will be white. So you end up with these really patchy looking roan cows. Another example is in chickens. You get checkered chickens. We call it checkered because literally one feather could be black and the one next to it is white. We get the same thing happening in flowers. So this would be incomplete dominance, where the red and the white combine to give you a pink offspring. This one's an example of codominance. Some of the cells are going to express the red pigment in its full red glory, and the ones next to it are going to express the white pigment in all of its glory. You don't get much pink bits. You get some red and some white. And this could be in really cool patterns depending on the expression. And remember, we've talked about epigenetics and how cells turn some genes on and some genes off so that they don't get expressed. Or it could be a wild mixture of cells, so you end up with this really spotty appearance happening. But this one is codominance. This one is incomplete dominance. Let's do an example. So if fur color is dominant in bears and we have a homozygous white male bear, So he would have big W for white fur. And it mates with a female black, so homozygous again, big B. What are the offsprings and the phenotypes and genotypes of the offspring? Well, we're going to have 100% big B, big W. They're all going to get one big W from dad and one big B from mum, and they're all going to be black and white. Now, I'm not saying that they'll all be pandas. This example just looks really cute because they look like pandas, but that's not how you get pandas. If you cross a black bear and a white bear, you don't get a panda bear. That's not how it goes. But you would end up with a spotty bear, where you would end up with some cells expressing the white pigment or the fur, and some cells next to it expressing the black pigment or fur. So, what are the phenotypes? It would be 100% black and white. There'd be a black and white bear. Whoops. And genotype would be 100% big B, big W, a heterozygous bear. Okay, your turn. Codominance. If a codominant checkered chicken, so remember checkered means that it's heterozygous, if that mother was mated with a true breeding, we're introducing some vocab here. True breeding means the same as homozygous. And that true breeding white chicken father and the mother who is checkered has an offspring. What's the probability of the offspring having A, all white feathers, B, all black feathers, and C, checkered phenotype? So pause the video here and have a go, and then we will go through the answers together. Okay, hopefully you've had a chance to do that. If not, pause me, rewind, and start again. So, father's genes. Father is a white chicken, and he is homozygous, so he is big W, big W. Mother is a checkered chicken, and those are codominant traits. So mother is a big B for black and big W for white. She's going to have some black and some white. So, this offspring would get a big B, big W, big B, big W. So we've got two checkered chickens straight out of the gate. W, 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 W. So all white feathers, what's the probability? Half of them are going to have all white feathers. So 50% is your probability of having all white feathers. Because these two here, the big W genotype, is going to result in white feathers. These are the homozygous white ones. All black feathers. Well, this one's going to be checkered, white, white. So 0% chance that from this particular combination of genes, we're going to get a black chicken. Not going to happen. And a checkered phenotype. We've got this one here and this one here. So that's 50%. So this question was only asking us for numbers. We were looking at probabilities only. Depending on how the wording of the question is phrased will determine whether you're looking for a number or whether you're actually looking for ratios. So read the question really carefully and always go back and check that you've answered the question properly. Another example of codominance is blood types in humans, and this is where it starts to get a little bit complicated. And I'm bringing some work forwards here. Remember back at the very, very beginning of our session, we talked about introducing polygenics, where there's more than two genes that control a trait. In this example, we have 3 alleles for the same gene. So we've only sort of talked about the fact that there would be 2 alleles for a gene, but it actually gets more complicated than that. Something like cat fur has up to 6 alleles of the same gene to code for different colors. Human blood type has 3 alleles. There's A, B, and O. A and B are codominant, so when the 2 of them come together, both of them are expressed. And O is recessive to both A and B. So if it's there with any of the others in a heterozygous form, O is hidden. O just doesn't get expressed at all. Now, these alleles code for an antigen on your red blood cells. And that has really important implications for who you can give blood to and who you can get blood from. Because if you have, let's say, B type blood and they inject an A type blood into you, your body is going to recognize it as a foreign substance and it destroy those blood cells. So there are 4 possible genotypes or blood types that are coded for by the different genes and the combinations that they can be found in. So if we think about it, we've got an A, so I could get 2 A's. I could get an A and an O. I could get 2 B's. I could get a B and an O. I could get an A and a B. Or I could get 2 O's. They're the different combinations that a human could get from their parents. So remember, one of these alleles comes from mum, the other one comes from dad. And these are going to code for different blood types in the end, different phenotypes. So you can have type A blood, type B blood, you can be blood type AB, or you can be blood type O. So blood type A can be achieved by inheriting an A from mum and an A from dad. In other words, you are homozygous for A. Or it can be determined by getting heterozygous, big A and an O. An O actually codes for no antigen at all being produced because it's recessive. You can get type B the same way. You can be homozygous, big B, or you can be big B, little o. There's only way to get type AB blood, and that is to get an A allele from mum or dad. Let's rephrase that. To get an allele from each parent that is either A or B, so that you end up with the AB combination genotype. And there's only one way to get type O blood, and that's to get both parents giving you or donating an O allele. Now, the interesting thing is that there is universal donors and there are universal receivers. Not all blood types are equal. So if you have type O blood down here, it means you produce no antigens on your red blood cells. So I could give your blood to anybody else, an A, a B, an AB, or an O. I could give it to anybody and their body won't reject it. So blood banks love type O blood. They're desperate for it because in an emergency, if they don't have time to check somebody's blood type, they just grab a bag of O blood because it can be given to absolutely anybody. Type O blood people are lifesavers in emergencies like car crashes and things like that. If you donate your blood and you're type O, they absolutely love you. If you are type A blood, you can receive blood from another A or an O. If you're a B, you can receive blood from a B or an O. If you're type AB, you are the universal receiver. Your blood, if you're in a car accident, you can receive blood from anybody and your body will not reject it. So you are effectively the genetic winners of this particular lottery. You're not going to be able to reject anybody's blood. We could just grab whatever and give it to you and you'd be perfectly okay. Your body's not going to reject it. Type O, however, while you are a universal donor and everyone loves your blood, unfortunately, you're a bit of a loser in life in terms of receiving blood because you could only receive type O blood. If I give you A, B, or AB blood, your body's going to attack it as a foreign body and you're going to probably die. So type O blood, it is actually in our best interest, and I'm a type O myself, it's in our best interest to actually give blood. Number one, because we can save all these other people, but also if you get in a car crash or you need a blood transfusion rapidly, you need O blood sitting there in the blood bank. You can't take anything else. So it may actually be your own donation that saves your life. So something to bear in mind. And this is why it all works. On an A-type blood cell, red blood cell, if you are an AO, it means that you have all these A antigens sticking off of it. If you have type B blood and you are a BO, it means you have all these B antigens sticking off it. And if you are an AB, it means you have both A and B antigens. So your body has gotten used to seeing these. This is normal. If it sees this come in, it goes, yeah, I know this. It's got A-type antigens. If it sees this one, it goes, yeah, I know that. It's got B-type antigens. So AB blood, and if you had an O here, it would have no antigens sticking off the outside whatsoever. So your body's not going to be able to recognize it, and therefore it would be accepted by an AB blood person as well. Your turn. Now, codominance is sometimes expressed, you probably saw back up here, that we sometimes express codominance as big A, and big A, little o. This one doesn't look like a little o, but it should be a little o. Or sometimes we express it with exponents, where we have I standing in for the allele, and then we have the A or the O representing what type of allele it is. So for this one, I want you to use exponents. So if a mother is heterozygous for blood type A, so blood type A, that means she is IA, IO. Doesn't look like an O. IA, IO. Heterozygous, two different ones. And her husband has blood type O. There's only one way to get O, and that is to have big I, big O. Sorry, little o, little o. It's the only way you could get it. So, pause the video here and see if you can solve it using exponents. It's the same process, you just got to remember to use the exponent notation. Okay, hopefully you've had a go at that. Let's see how you went. So, father was type O. So he's IO, off to work he goes. And mother is type A, but she's heterozygous, so she also has an O. So, this one's going to be A-type blood, heterozygous, AO. This one's also going to be AO. This one's going to get two little O's, and so is this one. So, what is the chances of the offspring having O-type blood? Well, that's this one and this one, so that's 50%. What's the chances of them having a heterozygous genotype? You've got to remember, a heterozygous genotype means that it has two different alleles. So that's this one here and this one here. So that's 50%. And what's the chances of it having B-type blood? Well, these two are going to be A-type, and these two are going to be O-type. None of them are going to be B-type. So chances of you getting a B-type child from that combination is 0%. None at all. Okay, have another go. This time we've got an AB mother has offspring with a heterozygous type A male. What's the probability of the offspring having type O, type AB, or type B blood? Up to you whether you use exponents or not here. I'm going to though, because I think you need the practice. Pause the video here and have a go before you keep watching. Okay, so let's go through. What have we got? We've got a mother with type AB blood. So she must be A, B. And a heterozygous type A male. Hetero meaning different. So he must be IA and IO. Shh. Sorry, my dog is snoring in the background. Oops. So, we have this potential offspring will be homozygous for A. This potential offspring will be heterozygous for A. This potential offspring will be AB. And this potential offspring will be heterozygous for B. Now you got to remember, I always put the codominant or the dominant allele first, and the little o always comes after, because he's recessive and he's going to get hidden. So what are the chances of them getting O-type blood? Well, there are 0% chance. There are none of them here that are going to have O-type blood. This one's A, this one's A, this one's AB, this one's B. So type AB blood. There's 1 out of 4. So that's 25%. It's only 1 out of 4 that's going to give you an AB. Type B blood, again, this one's A, this one's A, this one's AB, this one's B. So it's going to be 25%. The chances of them getting type A blood, 50%. Okay, the last thing we're going to go through today is a different form of inheritance again. And this time, it's not following Mendel's rules because the chromosome, the gene we're talking about is located on the sex chromosome, either the X or the Y chromosome. That means it's not going to be a Mendelian style of inheritance where one is dominant and one is recessive all the time. It probably is going to have that, but it's going to be inherited in a really strange way because remember that a male comes about because they get an X from mum and a Y chromosome from dad. But a female inherits an X from mum and an X from dad. So we have a different inheritance pattern in the chromosomes themselves. Therefore, we're going to have a difference in how the gene is actually inherited. And we'll start to see patterns in inheritance through generations to do with sex-linked traits. So that's really important that you remember that boys are XY and girls are XX. Well, I'm going to start with Y-linked inheritance. Why am I starting with Y? Because it's the easiest, but it's also the rarest. You very, 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 very rarely see this because the Y chromosome is tiny, really small. There just aren't that many genes on it. So the chances of you ever finding a sex-linked characteristic that is Y-based, almost not going to happen. You could almost skip this bit of the video entirely and not really worry about it because the chances of any question coming up on an exam that is Y-linked, almost nothing. Nobody ever really talks about it. But for the sake of gender equality, I am going to talk about Y-linked, sex-linked traits. So we know that boys inherit a Y chromosome from their father. So just a quick refresh on pedigrees here. You would have done this in year 10. The square ones are always males. You can remember that because the females are round and we're curvy and the males are squares, which of course they are. They're so much less cool than the girls. Don't actually mean that. I just use it as a way of remembering the squares are males, the round ones are females, okay? If it's coloured in, it means that it has inherited the gene that you're talking about. So if we're talking about a particular gene that, I don't know, causes a disease, and it's on that Y chromosome, this father is affected. He has an affected Y chromosome. So we show that with a little star next to his Y chromosome to say, this one's defective. He has a wife who is XX, and they have 2 children, a boy first and then a girl. So remember the order that they're in from left to right represents birth order. The male is of course affected because he's going to get an X from his mum and a Y from his dad. Dad only has one Y and it's faulty. It's affected. So he's going to give that Y to his son. And the pattern will repeat again with his son. And over and over and over again. Anything on the Y chromosome, every male is going to be affected. But this female, the daughter that they had second, is not affected because she doesn't inherit the Y chromosome. She inherits the X from her mum and dad's X chromosome. If she inherited the Y, she would be a he. Okay? So any female is never going to have sex-linked inheritance. So if you see a family tree that has nothing but males affected, it might be Y-linked inheritance. But you can't say that for sure without a DNA test because there are other ways, and sometimes it's just pure luck of getting every male in a family affected by a particular gene. Okay, so most of the traits are therefore X-linked. We say X-linked because they're on the X chromosome. So remember, this is a karyotype. We did this a few weeks ago, where you've got all the different chromosomes in a cell. And they should all be identical and paired up, except when you get to the sex chromosomes. In a female, they're identical. In a male, you've got one X chromosome and one teeny tiny little truncated Y. It's tiny, really is, compared to the X, it's ridiculously tiny. So we most often have changes in our X chromosome that get transferred on because the Y is just so little. How can you tell if a gene is sex-linked? Well, again, you can't conclusively prove it without a DNA test. But you can have a look at the family trees and see if you can see patterns, and you can start to make some assumptions there. Now, if we're just going to talk about X-linked for now, there are two different things that could happen, two different scenarios. There could be a gene on the X chromosome, and it could be a dominant allele. If this trait is dominant, then we have an affected mother and a non-affected father. She has 2 X chromosomes to pass on. One of them is affected, and it's dominant. He has 1 X and a Y. So the chances of this female being affected are about 50%. She's going to get one of these X chromosomes from dad, and it's going to be normal. And she's going to get one of her mum's X chromosomes. One of them is normal, and one of them is affected. If she gets mum's affected X chromosome, so there's a 50% chance there that that'll happen, she's going to have the disease. With the guys, the same thing happens, except that dad is volunteering a Y chromosome for the boys, which means it's all down to mum. He has a 50-50 chance that he's going to get mum's affected one or mum's normal one. If it's dominant though, it really doesn't matter whether he gets mum's dominant one, because there's nothing else to sort of counter it. So you can have a look at this family tree and you can go, well, every single female here is affected. And we also have a male in the middle. So there's a very good chance here that it's X-linked and that it's dominant, because lots of the females are getting it. They've got a 50% chance of getting it, but so do the males. They've got a 50% chance as well. It could just be purely coincidence that the females are getting it. This one, however, if we look at the recessive, we can see straight away that we have a lot of males in this generation that are affected. But it's not every male, because there is a female here. So this can't be Y-linked. It can't be on the Y chromosome. If it was on the Y chromosome, this female would never have inherited it. She would be a he. Okay, so we know that this female here must be big X, and this male must be XY. But because he's coloured in, he has inherited the faulty X chromosome gene or allele. He then passed it on to his daughter because he has only an X to give. The only one he can give her in order to make her XX is the faulty gene. So she's inherited it. He's giving the Y to his male son. Oops, that's not his son. That is somebody that she married. Now, this female here, she's affected. So she could be, if this is a dominant allele, she could be this genotype. She could be X normal, X affected, or she could be homozygous, X affected, X affected. Well, at this point, we don't know which. We know that she married somebody and had babies, and he is X normal Y. So, passing it on to all the boys in the family shows that the Y chromosome here is being given to each of the boys by dad. And every single one of these boys is getting an affected X chromosome from mum. Every single one of them. Which makes it very, very likely that she is the homozygous version of this, because every single boy has gotten the same one. We would expect to see about 50-50 of her boys if she is heterozygous. If she has one normal X chromosome, then it's 50-50 going off to her sons. But in this case, because she is the homozygous form, she's passing it on to all her kids. The fact that dad has an unaffected X chromosome and is giving that normal one to his girls saves them from being affected, because they're getting the affected one from mum, but because this one is recessive, it's hidden. They're carriers for the disease. They can then pass it on to their children, or their boys in particular, but they're not going to display the characteristics themselves. There's a really good video that shows you a lot about this that we're going to link onto our OneNote, so please make sure that you watch that. So just one more time, X-linked recessive inheritance. If we have the dominant, then we tend to see that because the father is unaffected, he's going to give his Y chromosome to each of his boys. And the mother has a good one and a bad one. So there's going to be a 50-50 chance for her boys, because her boys could get the good X or they could get the affected X. So what we're really looking at here is a Punnett square. So if we do mum, we've got XX, and mum has one good one, one bad one, and dad has one good X and a Y chromosome. If we get affected X, normal X, normal X, normal X, this one is an affected girl. This one is an unaffected girl. She's not going to have the disease at all. This boy is going to inherit it and get the Y from dad, and mum is going to give this boy a normal one. So for this one, you've got a 50% chance of having a boy and a 50% chance of having a girl. And within that, you've got a 50% chance of having an affected girl or an unaffected girl, and a 50% chance of having an affected son or an unaffected son. It's when you get to this side and you're talking about recessive inheritance that you sometimes find that those ratios change. So if you've got a carrier mother, one who has one normal gene and one affected gene, and you've got an unaffected father, if we do up our Punnett square, this time we get the same outcomes. We're going to get an unaffected and an affected. We get two normals. We're going to get an affected and a Y, and we're going to get a normal. So this one will end up with a 50-50 chance. It's when you get to the girls, though, that you've got a problem, because this one is dominant. This one is going to, if this one is recessive, this child is going to be normal, because she's going to get a normal X from dad. The unaffected one will dominate over the affected one. So all your girls are going to be normal. They're not going to be affected by this disease at all. So a 0% chance of an affected girl and a 50% chance of an affected boy. So recessive alleles on the X chromosome, you end up having a much higher chance of boys being affected, and therefore in a family tree you can see that a disease tends to strike the boys more often in a family than the girls. As soon as you see that pattern emerge, you've got to start thinking this might be a recessive allele that's being passed down by mothers who carry it hidden in their genome. They're not being affected by it because they're getting a good one from their father, a good copy of that chromosome, but as soon as the boys get it, they don't have that good copy from their father. They got a Y instead. They've got nothing to mask it, nothing to hide that recessive allele. So it tends to skip generations and will suddenly strike again the next generation in the boys. It's much less likely in the girls. In order to get an affected girl for a recessive, you actually have to have a father who is affected. So it's not like it's coming out of the blue. Nobody knew this was going to happen. Dad already has the disease. Things like hemophilia are X-linked recessive. Okay, that is all for today. I want you to go to the OneNote, please, and complete the activities that we have on there for you today. There's lots of practice ones to go through these. I think that's enough to go through today. And next lesson, we will go through lethal alleles, pleiotropy, and polygenetics.